Hello, everyone. Welcome to track 2. This is entitled, What's Next for mRNA? Glad you could join us. We're going to hear from 3 great speakers over the next 75 minutes or so, and then we'll bring them all back for a joint Q&A session for which we would love to hear your questions using the Q&A stage tab. Use the stage tab if you can see it. There's also a stage tab in the chat box if you just want to say hi, but the questions we'd like in the Q&A box. Coming up in this track, you're going to hear from, a little bit later, from Greg Newby from the Broad Institute. He's in the David Liu lab at the Broad, and Jaspreet Khurana from Strand Therapeutics. But first, I'd like to welcome Lior Zangi, Associate Professor at the Icahn School of Medicine at Mount Sinai in New York. Lior's lab focuses on modified mRNA, novel gene therapy delivery technologies, and their applications in changing the gene expression profile of the heart following myocardial infarction. Lior, welcome. Over to you.
Hello, and thank you for inviting me to give this talk. My name is Lior Zangi. I'm Associate Professor from Mount Sinai in New York. My lab focuses on the use of mRNA approach for treating ischemic heart disease. Ischemic heart disease is the leading cause of death in men and women in the world. Upon myocardial infarction, a heart attack is a blockage of the coronary artery and ischemic injury. This ischemic injury kills the cardiomyocyte, the contracting cells in the heart, and those cardiomyocytes are not being replaced by new cardiomyocyte, but rather by fibrosis and a scar. This scar prevents the heart to push back blood into the aorta. And there's no cure today. There's no any treatment that can create new cardiomyocyte and induce cardiac regeneration. However, we have avenues of research. There's tissue engineering when you use 3D scaffold to create a new muscle. There's cell therapy when we use iPS-derived cardiomyocyte or bone marrow-derived cells to inject into the heart to create new muscle and to create a better myeloid environment in the heart that will allow the heart to regenerate better. And there's the genetic medicine approach where we manipulate the protein expression in the heart and to stimulate proliferation of cardiomyocyte, in vivo reprogramming of non-cardiomyocyte to cardiomyocyte, or to enhance cardiomyocyte function. My lab works in the field of genetic medicine. Genetic medicine and gene delivery to the heart is basically to use tools to change the protein expression in the heart. As I said before, the heart have a very limited ability to regenerate itself. And we and others believe that if you change the protein expression in the key cells, you can change the heart from non-regenerative to regenerative ability, to have regenerative ability. And upon myocardial infarction, it will be able to regenerate itself. Frequently being used are AAV, mRNA, microRNA, and small molecules and protein. My lab focus on the use of mRNA. mRNA has been injected directly into the heart. There's no limitation in size. It doesn't compromise the DNA. The translation is being described for 3 days high, and then in addition 7 days with low expression. And you can control the amount that you put of mRNA. You can upregulate the gene by putting the open reading frame that translates your protein, or to use dominant negative approach if you want to downregulate a certain pathway, but they don't have any organ specificity or cell specificity. The use of mRNA was first described in the 90s by 2 papers in Science, showing that when you take an mRNA and you inject it to skeletal muscle onto the brain, you have a translation to a functional protein. However, this discovery was hampered by the ability of the mRNA to elicit an immune response. We know that mRNA that enter into the cell exogenously go into the endosome, and over there it's been recognized by toll-like receptors 7 and 8 that elicit an immune response. 2 researchers from UPenn, Dr. Kathy Carrico and Dr. Drew Wiseman, asked the fundamental question, why mRNA is immunogenic, and then identified that one of the ribonucleotides, uridine, has been recognized by toll-like receptors 7 and 8. In our body, we have over 30 types of uridine. One of them is pseudouridine. They nicely demonstrate that if you use pseudouridine, the toll-like receptor 7 and 8 do not recognize pseudouridine and do not elicit an immune response. That's make a new type of mRNA, they call it modified mRNA because it's modified in the uridine, to a tool that you can deliver mRNA into cells exogenously without the activation of the innate immune system. In addition, this change from uridine to pseudouridine also create protection from RNAs, so it was more stable, and increase the ADAS ribosome complex, which increased the translation of the mRNA, making an ideal tool for delivery of genes into cells. And in this COVID-19 pandemic, we have noticed the importance of this modification as the two vaccine, mRNA vaccine, and actually modified mRNA vaccine, as Moderna and BioNTech and Pfizer are using pseudouridine as in their mRNA vaccine. We in the lab use modified mRNA that we make ourselves. We use a plasmid, a DNA plasmid, and we clone in the open reading frame. We made over 500 genes like that. We use PCR to make a poly-A tail for this DNA. And this DNA we use for in vitro transcription with nucleotides that we buy from TriLink, the pseudouridine and the clean cap are from TriLink. And after the IVT, we clean the mRNA, and we can see using bioanalyzer that we have a very clean product. We use this mRNA and transfect it into cells to see translation. As you can see here, like Z with X-gal staining show a very nice translation of like Z. And our lab was really one of the goals of our lab was really to optimize this system, this modified mRNA, for delivery into the heart and into cardiomyocyte. And in the last decade, we were able to change the translation or increase the translation of the mRNA in the heart 20 times over. We did that by changing the pseudouridine that we are using, the cap that we have been using, the 5' UTR, the length of the poly-A tail. It's all been published by us, and those are the publications that describe what we have done to make this mRNA be more specific to the heart. We have an in vitro system where we transfect cells in vitro, and we also have an in vivo system where we inject the mRNA naked into the heart. As you can see here with LAG-C, the translation covers above 40% of the left ventricle, and it stays in the heart for 3 to 10 days of translation. In the last 7 years, our lab focused on 3 topics. One, as I mentioned before, is to optimize the mRNA for delivery to the heart. The second was to identify with this optimized platform new therapeutic genes. And we were able to identify 6 new therapeutic genes, and I'm going to talk about one of them today. The third, and you can see in green all the publications we have published in the last 4 years, 11 original research articles that covers those topics. And third, we also have the ability to overcome the lack of cell specificity, and we make the first cardiomyocyte-specific modified mRNA. And recently, we're able to overcome the need for open chest surgery. As I showed before, we inject the modified RNA directly to the heart, but now we show that we can do it through minimally invasive way through IV injection. But let me focus first on one of the targets, the therapeutic targets that we have shown in the heart. This work was over on direct reprogramming. Direct reprogramming of cardiac fibroblasts into cardiomyocyte was described first by Deepak Srivastava and then later by Eric Olson. They have shown that by using transcription factor like TBX5, GATA4, F2C, and Eric Olson added HEN2, you can transform fibroblasts to cardiomyocyte. In addition, you can enhance this reprogramming, direct reprogramming, by inhibit TGF-beta and Wnt pathway. The problem is that those genes were delivered in vitro and in vivo using retrovirus, and retrovirus increased the risk for cancer. In addition, those inhibitors of TGF-beta and Wnt were being used with small molecules, which increase the risk for side effects because the small molecules affect all the cells in the body, not just the cells that need to be reprogrammed. And we thought maybe modified mRNA could be a better approach. So in our study that was published recently in Molecular Therapy, we used alpha-myosin heavy chain mCherry mice. Alpha-myosin heavy chain is a specific gene for cardiomyocyte and basically makes all the cardiomyocyte in the heart red and cherry. And we use sorting to sort all the cells that are mCherry negative. And then we transfect them with GATA4, F2C, TBX5 modified mRNA. And we show that the modified RNA translates in vitro for about 3 days. So we decided to transfect every 3 days the cells and see if we transfect them with those transcription factor, which are GATA4, F2C, TBX5, HEN2, and dominant negative for TGF-beta and dominant negative for Wnt8. So you have inhibition TGF-beta and Wnt only in the cell that's going to be reprogrammed. And in addition, we add another gene, which is acid ceramidase that prevents apoptosis of the cell because all this reprogramming is have a lot of pressure on the cell. So it's 7 genes cocktail. And we wanted to know if you translate those 7 genes, we call it 7 genes cocktail, every 3 days, do you get reprogramming? And the answer after 14 days, we did see elevation in alpha-actinin, which is a specific marker for cardiomyocyte. However, when we wait for day 21 and day 28, we found out that the number of cardiomyocytes that have been created are insignificant. It's like 2 out of 50,000. It's not something that can really make a difference. When these 7 genes were injected in vivo in a myocardial infarction model, and we waited 28 days, we saw an improvement in injection fraction, reduction in scar size, reduction in collagen and fibronectin production, and even long-term survival for over 6 months after a myocardial infarction. And then we thought maybe in vitro we couldn't create cardiomyocyte, functional cardiomyocyte, but maybe in vivo we can create functional cardiomyocyte. So we tested that by linear tracing model. In this linear tracing model, we gave tamoxifen before myocardial infarction. This tamoxifen marked all the pre-exist cardiomyocyte, and then we inject those 7 genes, we wait 28 days, we collect the heart, we make slides to in immunostaining for cardiomyocyte, pre-exist, and new cardiomyocyte. And those new cardiomyocyte is here in purple, and one can see that this is not mature cardiomyocyte. It doesn't even look like cardiomyocyte. But somehow, they are like 25% of the left ventricle of those cells. Somehow those cells are doing good for the heart after myocardial infarction. And we wanted to know how. to understand better, we use a single cell RNA analysis for cells that got the 7 genes and not. And you can see that these 7 genes mRNA translate very nicely in the same population of cells. And when you look, you use Go analysis to see what kind of a structure, biological processes are going in those cells. You see that there's cardiomyocyte process, cardiogenesis process, and angiogenesis process. However, please remember that 4 of the transcription factor were being upregulated by us, GATA4, F2C, TPX5, HEN2. So then we wanted to exclude the 7 genes and then just measure what biological processes are going in the cell. And the only one, all the muscle kind disappeared, and the only one that stayed was angiogenesis. Further investigation into the cluster analysis of the cell, and then they find one cluster that was very low on the control Lutz-treated group versus the 7 genes treated group, which is a population that enrich in processes of angiogenesis, VEGF, endothelial cells proliferation, VEGF signaling, and upregulate a marker like SCA1 and CCL2, which are marker for mesenchymal stromal cells. Also, the transcription factor that have been upregulated are known to be mesenchymal stromal cell pro-angiogenic markers. So we call those new population that it's like 40% of the treated cells 7 gene-induced mesenchymal stromal cells. And because those mesenchymal stromal cells really induced angiogenesis, we went back to the treated heart, and we indeed saw an elevation in capillary density after injection of 7 genes in the cardiac tissue. And when you isolate cardiofibroblasts and cardiomyocyte, you give them both 7 genes, you see that the cardiac fibroblasts actually upregulate almost a high amount, more than 10 different angiogenic factors. To show that this is really angiogenic and not a cardiac-related phenomena, we did the same experiment with ischemic injury in the ischemic limb. And over here, we also saw upregulation of angiogenesis and increase in capillary density and upregulation of many angiogenic factors, showing that those genes, those 7 genes, are not really directly reprogram cells to cardiomyocyte, because we don't have cardiomyocyte really in skeletal muscle. The whole idea and the way they benefit is actually by inducing angiogenesis and upregulate pro-angiogenic genes. So we describe that 7 genes basically promote a better vascular niche to induce angiogenesis, but not cardiomyogenesis. So there's no new cells that being formed, new cardiomyocytes that are being formed, but actually the niche of the vascular niche is being better. And this is by direct injection of those 7 genes that induce cardiomyocyte-like cells, which are 7 genes induce MSCs that can secrete many angiogenic factors that promote angiogenesis and reduce fibrosis in the heart and in the ischemic leg. Now we are moving forward with our study into pigs. We have a collaboration with Jay Zhang from the University of Alabama and Hisham Sadek, where we use those therapeutic genes that we describe in our papers, and welcome you to go and read our new target that we describe, and trying to identify and to evaluate how those therapeutic target works in large animals, in pigs. So in here, just as a proof of principle, we created LAD, and we gave an injection again of LAG-Z modified mRNA. And one can see very clearly the ischemic area. This is the white area in the heart that did not been transfected, but it's surrounded by healthy tissue that were transfected well with the 7-gene modRNA, with the LAG-Z modRNA. And now we want to use 7 genes and other therapeutic targets that we describe to trying to reduce the scar formation in pigs. In the last few minutes that I have for my talk, I would like also to mention another new things that we are working on, which is to reduce the need of open chest surgery. We described in 2017 that when you directly inject modified RNA to the heart naked, you have expression only in the heart and not in other cell type, other organs. And now we show that if you capsulate the modRNA and you can make it more cell-specific to express only in cardiomyocyte, you can inject it IV, but the translation will be only in the heart in cardiomyocyte. You can see over here what happened when you give a IV injection of modified mRNA regular. And you can see that the translation is in all organs, in the heart, in the spleen, in the lung, and in the liver. However, we created a system that we call SMART, a specific modified RNA translational system that based on 2 mRNA, that one is the suppressor, one is the gene. And so there is no translation. And upon putting recognition site on the suppressor gene, we eliminate the suppressor gene in a specific cell type, in this case in cardiomyocyte, and allow the translation of the modRNA only in cardiomyocyte, and cardiomyocyte we have only in the heart. So one can see very clearly that the mRNA translate solely in the heart after IV injection. We have shown that if you give an IV injection every 3 days, you can replenish the mRNA translation in the heart, and using mCherry cardiac SMART, you can find that the translation of the mCherry is only in cardiomyocyte, but not in other cell type, which is make it extremely specific to the heart and to cardiomyocyte. We compared what happened when we deliver this IV injection after heart attack in mice when we use Lutz control or therapeutic cardiac SMART, one of our genes, and we gave this 4 injection every 3 days for 9 days. So basically day 0, day 3, day 6, and day 9 after a heart attack. And we measure echo and tunnel staining. And you can see 48 hours after IR, you see a high reduction of tunnel positive cells. So our therapeutic genes can give a protection of the myocardium and reduce tunnel cell death in the heart significantly. This result in day 28 in a reduction in the scar size. And less scar size, you have an improved cardiac function 28 and 29 days later post a myocardial infarction. This is the first demonstration that I have seen of an injection of modified mRNA IV that can change and protect the heart post a myocardial infarction and reduce remodeling in the heart post myocardial infarction. And it's only by 4 IV injection in the first 10 days. I would like to thank my labs, Kirat Kaur, Magdalena Jacques, Anna Nukurin, Jamin Yu, and Seong Yu, and the former lab members, Ajit, Nishat, and Yoav. Also our collaborators, Jay Zhang and Hisham Sadek, and our funding, of course, from industry and from NIH, and for you for coming to my talk. If there's any question, I'll be happy to answer.
Okay, our next speaker here in track 2, What's Next for mRNA, is Greg Newby, a postdoctoral fellow in the David Liu Lab at the Broad Institute and Harvard University. Greg was a graduate student with the late Susan Lindquist at MIT, where he developed methods to track and modulate protein aggregation and yeast prions. For the past few years, he's been a postdoc in the rather productive Liu Lab at the Broad, developing therapeutic applications of base editors and prime editors. Greg is going to discuss base editing towards a therapeutic for sickle cell disease. Greg, really looking forward to your presentation. Over to you.
Hi, I'd like to thank the organizers for inviting me to talk to you today about our work using base editing towards a therapeutic for sickle cell disease. I'm a postdoctoral fellow in the lab of David Liu, and much of our lab is focused on treating human genetic disease. There are over 75,000 different pathogenic genetic variants. About half of these are caused by single nucleotide polymorphisms, which are indicated on this pie chart by the blue and red slices. Cas9 nuclease has really revolutionized our ability to target many parts of the genome for modification, but it's not very efficient at installing single base changes in a large majority of the cellular population. When we target a nuclease such as Cas9 to a genomic site, it will introduce a double-stranded DNA break, and generally these are repaired by cellular DNA repair machinery, leading to a small insertion or deletion at that targeted site in the genome. It's possible to use HDR, homology-directed repair, and encourage cells to encode a precise edit based off of a donor DNA template that we provide. However, this process is most efficient in quickly dividing cells, and most cells in a human or an animal model are slowly dividing or not dividing at all. So HDR is generally inefficient. Of the 43 clinical trials using CRISPR technology that were registered earlier this year, most of them were involving Cas9 to do gene knockouts, and there were none of these clinical trials that were focused on direct SNP correction to treat genetic disease. To address this unmet need, my predecessors and colleagues in the Liu Lab, Alexis and Nicole, developed two classes of genome editors, that they call base editors, that make C to T or A to G changes in the genome. And together, these can correct the 4 types of mutations, the transition mutations that make up this largest slice of the pie of all human genetic disease variants. For the sickle cell work, the base editor we made use of is the A base editor. The A base editor is pretty special because it can correct nearly half of all of the disease-causing single nucleotide changes. It does this by using an enzyme to conduct this chemical reaction directly in the genome of the cell at a targeted nucleotide. We'll deaminate the adenosine and generate an inosine instead. This inosine is read as a guanine by cellular DNA repair machinery and polymerases. The A base editor enzyme is composed of these two components fused together, one being a Cas9 nickase that contains a mutation that disrupts the double-stranded DNA cleavage activity so that Cas9 can make just a nick in the DNA, and that's fused to a deoxyadenosine deaminase that conducts this chemical reaction. So after the base editor binds to the target site in the genome, the protospacer anneals to a complementary sequence in the genomic DNA, and this displaces one of the strands of DNA. The A's that are within a short window of this displaced R loop are accessible for deamination and base editing. So following the activity of the deaminase turning A to I and activity of the nickase nicking that opposite strand, we encourage DNA repair to preferentially replace this bound strand by the protospacer and install the edit. My main work in the Lu Lab has been to apply this tool to treat sickle cell disease models. Sickle cell disease is the most common deadly genetic disorder in the world, and it's caused by an A to T transversion mutation in the hemoglobin beta gene. This results in polymerization of deoxygenated hemoglobin and leads to organ failure, pain, and early death in patients. Most patients do not have a suitable donor for allogeneic bone marrow transplantation. Therefore, many strategies that are currently being pursued in the clinic make use of autologous stem cell modification followed by retransplantation back into the patient. To modify a patient's own cells, several strategies make use of viral vectors to deliver therapeutic constructs, or the use of nucleases to de-repress fetal hemoglobin or stimulate homology-directed repair. However, we envision developing an improved therapeutic that doesn't involve the delivery of DNA through a viral vector or the double-stranded DNA breaks that are caused by nucleases in order to minimize the genotoxic events. In order to develop base editors that can efficiently modify the sickle cell disease-causing mutation, we used phage-assisted continuous evolution, or PACE, which is a directed evolution platform developed in the lab of my mentor, David Liu. To conduct PACE, we encode the gene that we wish to evolve in a bacteriophage, and we set up a circuit in a bacterial strain so that activity of that gene is linked to greater expression of gene 3, which is essential for the propagation of the bacteriophage. We can conduct many, many rounds of evolution all over the course of a few days without much intervention by the experimenter, just by continually growing a co-culture of the phage and bacteria in a turbidostat. Phage that contain very active genes will propagate rapidly and will be maintained in the culture, whereas phage that encode inactive genes will be washed out of the culture quickly. And in work led by my lab mates Michelle Richter and Kevin Zhao, we encoded a genetic circuit that will activate gene 3 in response to A base editing by installing 2 stop codons into T7 RNA polymerase. Only after the A base editor successfully removes those stop codons do we get expression of full-length T7 RNA polymerase, which can then transcribe gene 3. The resulting deaminase, called ABE8E, is over 600-fold more active than the original deaminase ABE710. When we assess this deaminase in a base editor in human cells, we see substantial improvements to activity. The original ABE710 base editor is shown here in gray, and at some sites, the activity is not much improved by the new base editor shown in red, but at others that are poorly edited, like the sickle cell site was at first, we get substantial improvements using ABE8E. The other avenue of improvement that we needed to make was to actually permit the Cas9 to target the sickle cell site. And that's because there's no NGG PAM that normal wild-type SP-Cas9 likes to use located within the right position relative to the sickle cell mutation. Therefore, we again use this phage-assisted continuous evolution platform. This time, instead of evolving the deaminase, we evolved the Cas9 variant so that only Cas9 variants that bound to a target site located upstream of gene 3 are able to recruit RNA polymerase and transcribe that necessary factor. Following substantial evolution efforts, we eventually isolated 3 variants known as NRRH, NRTH, and NRCH which are the respective PAMs that these variants recognize. Here we're listing the new mutations that were introduced in each of these variants and led to improved targeting for those PAMs. So this took us from being able to access around 25% of sites to now being able to access around 95% of sites. And thankfully, one of the sites that is now targetable is the sickle cell locus. We combine these two engineered domains to produce a base editor that's custom tailored to modify the sickle cell target site very efficiently. We use this editing strategy, the protospacer here underlined in green and a PAM underlined in purple, which is CACC PAM sequence. This places the target sickle cell causative mutation at position 7 of the protospacer, and editing it converts the toxic valine into an alanine amino acid. While this isn't the wild type amino acid at this position, it is a naturally occurring variant known as hemoglobin Makassar, which is non-pathogenic. Other bystander edits can occur at positions 9 and 12. Both of these would lead to only silent changes. However, a bystander at position 16 would lead to an undesired coding change. We electroporated this base editor as either an mRNA or an RNP into hematopoietic stem and progenitor cells from patients. We observed up to 80% correction of the target site, while the non-synonymous bystander mutation, which would be a red bar here, was not observed. Fewer than 3% of alleles contained an indel in these edited cells. And we saw that mRNA generally did a better job of editing human cells relative to RNP. We differentiated these HSPCs into erythroid cells, into red blood cell progenitors. And here we're measuring, using HPLC, what the globin content is. The gray bars here indicate the toxic sickle globin, whereas blue bar is our desired Makassar globin. We see the corresponding shift relative to the editing frequency of sickle globin into Makassar globin. When we placed these erythroid cells under hypoxia, we saw a substantial reduction in the propensity of these cells to sickle. We assessed off-target editing in response to base editor treatment using two methods. First, we nominated sites using Cas-Off-Finder, a computational search for those regions in the genome that are most similar to the protospacer, and also nominated other sites using CircleSeq, an experimental method that looks for where Cas9 complexed with the same guide will cut. We sequenced nearly 700 of these potential sites and identified verified off-target editing at 54 of the sites. For the most part, these off-target sites were edited with low efficiency of around 5% or less, but a few of these sites were edited efficiently, even up to 80% editing. Looking at the annotations of these regions, they largely fell in intronic or intergenic parts of the genome. 4 of them did fall in exons, but all led to silent mutations. A small number fell close to regulatory regions. However, each of these were still several hundred bases away from the nearest exon, so we don't anticipate any off-target edits of clinical relevance. We also looked at some of the other phenotypes that have been reported to occur after Cas9 treatment. We also assessed whether base editing resulted in some of the same undesired outcomes that Cas9 nucleus has been reported to result in cells. One of these is the p53-mediated DNA damage response, which can lead to reduced cell growth and fitness. To measure this, we looked at transcript levels of p21, which is a target of that DNA damage response. We saw that, as previously reported, treatment with Cas9 nucleus does substantially increase this response, and it's sustained for over 48 hours. In contrast, cells that were treated with base editor mRNA did not show any induction of this response relative to controls that were electroporated with no cargo. Another undesired response, another undesired outcome of Cas9 cutting is loss of substantial regions of sequence that are surrounding the target site that could range from hundreds to thousands of bases. This is difficult to quantify by traditional amplicon-based sequencing because the primer binding sites to conduct PCR for analysis are no longer there. So to quantify the loss of these target sites, we use droplet digital PCR. We observed that cells treated with base editors showed no substantial change in the content of that target site following treatment. In contrast, cells that were electroporated with Cas9 nuclease did lose around 14% of the sequence surrounding the target cut site. So Based on these outcomes, it seems that base editing can be less perturbative to the cell. We next asked whether the cellular population that we're editing in those HSPCs is including the long-term bone marrow repopulating hematopoietic stem cells that would be necessary to treat the disease in a patient. To answer this question, we transplanted our edited cells into immunocompromised NBSGW mice. And 16 weeks following transplantation, we harvested the bone marrow for analysis. The idea is that if bone marrow repopulating cells were truly edited, they would persist throughout the 16-week period, and we would still observe them at the end. Whereas if we had only edited progenitor cells that rapidly differentiate and are lost, then we would see a significant drop in editing. We saw that our editor treatment did not lead to any sort of perturbation in those stem cells, in that engraftment was the same in edited or unedited control cells. Also, the lineage potential of these cells to differentiate into T cells, myeloid cells, or B cells was not altered by treatment. Erythroid cells are also quantified here. When we isolated the cell populations that we can enrich by flow cytometry and conducted amplicon sequencing of the target site, we observed an average of 68% allele frequency of our Makassar, the desired Makassar allele, about 68% of those alleles. This indicated to us that we had edited approximately 68% of the alleles in those bone marrow repopulating stem cells. We conducted single-cell RNA-seq to determine the editing outcomes, whether they were monoallelic, receiving a copy in only one beta-globin allele, or biallelic, being edited in both. We saw that approximately half of the cells were monoallelic and half were biallelically edited, but about 80 to 90% of the cells received at least one edit. And all of those cells with at least one edit we expect to be non-pathogenic anymore. So this was a very exciting result to us. Using HPLC to assess globin content, we again saw this shift of about 60% of the sickle globin into Makassar globin. And placing these cells under hypoxia, we again observed a substantial drop in the propensity of the cells to sickle. All of these point to the treatment reducing the pathogenic potential of the sickle cell mutation, restoring cells to a healthy state. The NBSGW mouse can determine whether these cells are bone marrow repopulating, but it can't tell us whether we're really treating the disease phenotypes in the mouse, because human cells don't persist long in the mouse periphery. So to answer this question, we used a mouse model that contains human hemoglobin beta allele and a few other human alleles as well. And these mice actually show the symptoms of sickle cell disease. There are disease mice with 2 copies of the sickle cell mutation, and there are also control mice that have a healthy human allele. In this case, we're using a heterozygote that contains 1 healthy allele and 1 sickle allele as a healthy control. So to figure out whether we can conduct a bone marrow transplantation with edited cells and treat this disease, we extracted Lin minus hematopoietic stem and progenitor cells from these mice. We electroporated those from the sickle model with A-BAIT-E and RCH to modify the sickle allele to Makassar. We also conducted an untreated control. So these animals received cells that should remain as biallelic sickle cell allele cells. And then we also did not treat our healthy control. And 16 weeks after transplantation, we assessed all of their phenotypes alongside a non-transplanted healthy control mouse indicated in brown. We saw that before and after transplantation, during that 16-week period, we maintained around 40 to 50% of the Makassar allele. In this case, HPLC assessment of the blood indicated that we were getting 70 to 80% Makassar globin content. We think that this is happening because cells that have been treated are no longer sickling, and therefore they're less delicate, and they persist longer in the blood, making up a larger fraction of the hemoglobin. We measured several hematological parameters of these mice and observed that in each case, the treated mice that are indicated here by with the blue bar appeared very similar to the healthy controls and different from the mice receiving untreated cells indicated by red bars. This is true for both the total hemoglobin content of these mice, of the blood, the percentage of reticulocytes, the count of red blood cells, and the count of white blood cells in these animals. We placed blood from these mice under hypoxia to assess sickling and observed again that those mice that received edited cells have a greatly reduced propensity to sickle, which is quantified here at right. We also used a colony forming assay to assess whether the editing outcomes were monoallelic or biallelic. And again, we observed that it's approximately 50-50 split between the two. And in this case, around 80% of cells received at least one edit. We looked at spleen pathology to assess the mutant phenotype and the effective correction in this case. The red pulp of the spleen is generally expanded in mice that receive sickle cells because there's a lot of erythropoiesis that occurs outside of the bone marrow in the spleen in order to make up for the lack of red blood cells in the, due to the delicacy of those that sickle. Mice that received our edited cells show a greatly reduced red pulp relative to disease cells, and they look much more similar to healthy controls. Due to pooling of blood in the spleen, those with the sickle cell model are greatly enlarged, a splenomegaly phenotype. This is also reduced in animals receiving our edited cells by about 60%, quantified at right. Lastly, we conducted a secondary transplantation to see whether the edited cells were really persistent over a second 16-week period, and also to get a sense for what allele frequency is necessary to conduct to achieve phenotypic correction. So we took bone marrow from one of these mice receiving treated cells that had a 39% hemoglobin Makassar allele frequency at the time it was sacrificed, and we mixed these in 6 different ratios with bone marrow from a mouse transplanted with unedited sickle cells. At each of these ratios of healthy to unhealthy bone marrow, we transplanted them into 3 different new mice. We assessed their phenotype 16 weeks after the secondary transplantation. We observed that the frequency of the hemoglobin Makassar allele by high-throughput sequencing was stepped in accordance to what we'd expect based on the ratio we transplanted. We saw that in all of the phenotypes of disease, including the hemoglobin level, percent reticulocytes, red blood cell count, and white blood cell count, that an allele frequency of about 20% noted in light blue is about sufficient to restore these mice to the level of a healthy mouse. The healthy control mice in this case are indicated by the dashed lines. The result was really encouraging to us because in human cells, we were able to achieve 68% allelic correction. So we think that that's in far excess of what's necessary to actually correct a human patient and treat the disease. In summary, we saw that base editor mRNAs can convert the sickle cell allele to a benign variant in bone marrow repopulating hematopoietic stem cells from sickle cell patients. We also saw that the same editing strategy can improve disease parameters in sickle cell mice, and that this could serve as the basis of a potential autologous therapeutic that eliminates the pathogenic mutation and protein and avoids the undesired consequences of double-stranded DNA breaks. With that, I'd like to thank all of our collaborators, especially Jonathan Yen, Caitley Woodard, and Thiagaraj Meyer Nathan, who led this study with me. And I'd also like to thank Mitch Weiss and David Liu, who were the primary supervisors for the study, as well as all the funding sources listed below. Unfortunately, I'm not able to attend the question and answer session due to a prior commitment, but please feel free to send any questions to this email address here, and I'd be happy to discuss with you. Thank you all for your attention.
Wow. Thank you, Greg, for a great talk. If you have a question for Greg or Lior or our final speaker coming up, please enter them in the Q&A stage tab and we'll get to them shortly. All right. So rounding out track 2, what's next for mRNA is Jaspreet Khurana, who is Director of mRNA Programming at Strand Therapeutics. Jaspreet is a molecular biologist specializing in mRNA therapeutics and genome engineering. He took his PhD from UMass Medical School, followed by postdocs at Princeton and Columbia before moving into industry. He joined Strand about 2.5 years ago and is currently Director of Platform and Discovery. He's going to talk about programming mRNAs for cancer immunotherapy.
Hello, everyone. My name is Jaspreet Khurana, and I'm the Director of mRNA Programming at Strand Therapeutics. I want to thank the organizers for giving me this opportunity to present at the annual mRNA Day. So thanks to TriLink, the CRISPR Journal, and the Genetic Engineering and Biotechnology News Network. And this is my disclosure. Strand Therapeutics is an early stage mRNA therapeutics company based in the greater Boston area with the founding technology out of MIT. We have published our results in high-profile peer-reviewed journals. And earlier this summer, we raised $52 million in Series A funding with participation from Redmile, Playground, Andri, and others. Some of our key achievements since inception include getting an exclusive license of our foundational MIT IP. We have nominated our first lead development candidate, STX-001, a self-replicating mRNA targeting solid tumors. And we have inked $277 billion Asia licensing deal with Beijing. So in this talk, I'll present a general mRNA landscape in the clinic, and then move on to describe specifically the role of mRNA drugs in the cancer immunotherapy landscape. And then finally, talk about what we at Strand believe could be the future of mRNA in cancer immunotherapy. While mRNA has been an important subject matter of research for the longest time, its role as a gene therapy vector was only realized back in 1990, when John Wolfe, Robert Malone, and colleagues first demonstrated protein expression using a synthetic mRNA in the mouse muscle. But it wasn't until later in 2005 when Carolyn Carrico and Drew Weissman out of UPenn demonstrated that compared to an unmodified, a natural mRNA, a synthetic mRNA made using base modification not only provides improved protein expression, but it does so by evading the innate immune stimulation. And this much needed molecular insights into the mRNA biology further led to rekindled interest in mRNA as a therapeutic modality, later in the decade leading to inception of mRNA companies like BioNTech and Moderna. In 2011, DARPA realized the need for getting ready for pandemic responses and led $300 million funding to work on nucleic acid technologies, including mRNAs. Later in that decade, our co-founders started Strand out of MIT with the promise of building the next generation of mRNA medicines. In 2020, mRNA really took the center stage when the COVID vaccines from BioNTech and Moderna showed remarkable efficacies in clinical trials, leading to the first FDA approval of mRNA drugs earlier this year, saving millions of lives. In 2021, earlier this year, the momentum for mRNA continued with Intellia successfully reporting gene editing of the TTR gene in patients for their in vivo gene editing program using Cas9 mRNA. So some say that we are headed in this decade of mRNA therapeutics. So much so that there has been a massive pharma investment in the mRNA space where big pharmaceutical companies are trying to capitalize on this newly de-risked modality. So there have been multiple reports from pharmas like Pfizer, Novartis, Sanofi, to name a few, who are trying to either internalize their own mRNA technology or create mRNA programs by mergers and acquisitions. And this is the mRNA landscape in the clinic. As is evident, there really has been an explosion of activity in the mRNA space in the recent years. There are more than 68 companies developing mRNA drugs. And in this graph, the size of the bubble reflects the number of mRNA drugs led by different companies in various stages of the pipeline. There are more than 200 mRNA drugs in the pipeline and more than 250 clinical trials which have been initiated to date. Further breaking them down by therapeutic area and clinical trial phase, it is evident that the therapeutic area in which the most mRNA drugs are being tested is that of infectious diseases, so vaccine stage. But this is primarily led by studies being in the preclinical stage. But if we look at the top therapeutic area by number of mRNA drugs in the clinic, that is led by cancer, which is 48%. So now let's talk about the specific mRNA oncology landscape. And why mRNA? I mean, mRNA, I think, is an ideal gene therapy vector for cancer therapy. Not only does it provide extra safety not seen in the viral vectors, but at the same time, it has the ability to deliver large payloads, is easier to deliver systemically, and is easily scalable as well. If you look at the number of mRNA drugs by clinical indication, there are 75 mRNA drugs in oncology currently. More 90% of them are addressing solid tumors, while 10% address hematological malignancies. And even though close to 60% of these are in preclinical stages, there are a handful of them in different stages of clinical development as well, with top cancer indications including non-small cell lung cancer, ovarian cancer, and melanoma. What has been interesting to see over the years is the evolution of the mechanisms of action by which these different mRNA cancer drug programs, what they have been utilizing. So earlier in the decade, while primarily the mRNA cancer drugs were targeting cancer vaccines, in more recent years, there has been more clinical trials using mRNA as a gene therapy vector for driving cytokine or antibody expression, as well as personalized cancer vaccines. This is excluding any adoptive T-cell therapies. And if we further break this down to understand the different payloads that have been used in these different mechanisms of action, cancer vaccines still are the top mechanism of action, riding at 39%. And within them, within the different mechanisms, we have the major payloads include neoantigens, cancer antigens, including HPV, cytokines like IL-12 and IL-2, and various antibodies. The immune system has a complicated role in tumor progression. While it is well known that the immune system can restrict tumor progression, it can also lead to tumor progression, as has been seen in not only preclinical studies, but also reports from clinical studies as well. So according to this cancer immunoediting model, there are 3 phases in tumor progression. So in the beginning, the effector cells from innate and adaptive immune system try to eliminate any transformed cells in the body. And while this process is mostly very successful, there are some cells that escape this elimination and can lead to the next stage, in which the tumor cells are largely dormant and reach an equilibrium stage, where a constant pressure from the adaptive immune system combined with the genetic instability of tumor cells leads to selection of those rare tumor subclonal populations, which are now immunoedited and can really escape and lead to a full-blown tumor progression when the tumor becomes clinically apparent. Some examples of these tumor immunoediting include PD-L1 depletion. And given this heterogeneity in the process and what we see in different tumors within the clinic, it has been difficult to deploy mechanisms like checkpoint inhibitors, for example, which have been known to work for and are approved in multiple different indications. The problem is that the response rates are generally modest. So there's clearly a need to deploy multiple mechanisms to target tumor progression. And luckily, that's what we have been seeing in multiple clinical trials. So this is an overlay of different oncology mRNA clinical trials with readouts overlaid on the infamous Chen and Mellman cancer immunity cycle. So what has been promising so far is that all these mRNA trials in oncology have been well tolerated. 70% of them are cancer vaccines. 60% have used multiple mRNAs. And the different programs are targeting different sites in the immunity cycle, including cancer antigen presentation, priming and activation of T cells, or recognition of cancer cells by T cells. Unfortunately, only a handful of trials so far have reported promising reports. But I do want to highlight one example of a clinical trial which has shown promising phase 1 results. This is the MEDI 1191, an AstraZeneca Moderna sponsored trial, which includes an intratumoral delivery of a lipid nanoparticle containing a modified mRNA expressing IL-12. IL-12 is a T and NK cell activation chemokine, which leads to production of interferon gamma, which not only leads to T cell recruitment and activation, but also PD-L1 expression. On the right is the clinical readout from a patient showing a 61% reduction in tumor size in non-injected lesions in a metastatic mucosal melanoma. These are highlighted by the yellow arrows, and so far they have shown efficacy, and the trial has a reported 20% ORR. But clearly we think that we need to do better in the cancer immunotherapy space. So we at Strand believe that we are trying to build the future of mRNA in cancer immunotherapy. And we do that by utilizing the two cornerstones of the Strand platform. The first involves expression from a self-replicating mRNA. While a conventional modRNA has a lifespan measured in days, a self-replicating mRNA derived from an RNA virus not only provides a massive improvement in protein expression, but also a more sustained expression within the body. We have demonstrated the efficacy of driving expression by a self-replicating RNA. This is studied by our MIT co-founders who found that an oncolytic LNP containing a self-replicating mRNA driving IL-12 expression not only leads to immunogenic cell death and T cell activation, but also primes systemic immunity within preclinical models. This is data showing that a single dose of LNP containing replicating RNA expressing IL-12 provides a curative single agent activity in an immunologically cold tumor model of melanoma called B16F10. So they demonstrated that compared to the controls, LNP containing IL-12 replicating RNA leads to a dramatic tumor shrinkage as well as improved survival of the treated mice. On top of that, the B16F10 melanoma model is a PD-1 refractory model. So what they demonstrated in that study is that combining anti-PD-1 with our replicating mRNA expressing IL-12 leads to a more synergistic effect in tumor shrinkage, which is very promising. The second cornerstone of the STRAND platform is our ability to add control to our mRNA drugs. So we do that by making our mRNAs cell type specific, and we can also tune the expression level of the proteins from our mRNAs. This not only helps us avoid off-target effects that may arise from side effects of expression in the off-target tissues, but we can also provide the desired protein levels for therapeutic proteins with a narrow therapeutic index. So we at Strand believe that programming mRNA is very much akin to a programming language. So if our mRNAs can sense a disease state, they can activate multiple mechanisms. If, on the other hand, they sense a healthy state, they can get degraded to avoid those off-target effects. So we use the principles of synthetic biology in our gene therapy programs. So our mRNAs can sense either external regulatory signals or intrinsic biomarkers, perform logic processing within the cell, leading to a more regulated therapeutic function at the end. To give you two examples of this platform, in one scenario, we have demonstrated that a microRNA sensing mRNA program can be utilized for a cancer-specific protein expression. MicroRNAs, as some of you know, are small non-coding RNAs involved in eukaryotic translational repression. In our case, we are using them as cell type specific biomarkers. So in a co-culture of cancer and healthy cells, we are trying to drive expression of an apoptotic factor called BAX, leading to apoptosis specifically of the cancer cells while sparing the healthy cells. And we do that by adding a cancer cell low microRNA sensors within the payload strand. We add a further control by regulating the expression of this BAX protein by an RNA binding protein, which can downregulate the expression of this in healthy cells. And we regulate the expression of this repressor by encoding a cancer cell specific microRNA miR-21. So when this circuit is transfected in this co-culture setting, we see that upon this program, circuit can specifically lead to cancer cell death while sparing the healthy cells away. In a second scenario, we have demonstrated that we can utilize a small molecule-based adjustment of protein expression. So in this case, we have shown a dose-dependent effect of a small molecule like trimethoprim in creating an off switch for the payload of interest, leading to more than a 20% decrease in payload expression in a dose-dependent manner. So the STRAND mRNA platform really is a plug-and-play strategy where we combine our indication-specific targets with our modular platform to create these genetic circuits, which can then be utilized for specific indications, providing tissue type specificity, leading to production of a modular combination of drugs targeting many different indications. So in summary, I've told you today about the history as well as the general landscape of the different mRNA drugs in the clinic, focusing on oncology and showing that there have been at least a few promising clinical trial readouts. Safety so far has been really good, regardless of the modality or delivery or mechanism of action. So the future of mRNA looks promising in oncology, and we at Strand believe that it really could involve the precise programming of protein expression using design principles from synthetic biology. So with that, I want to stop and acknowledge really all my colleagues at Strand, especially folks from the BD and data analytics team who helped provide the slides for the mRNA landscape, along with our MIT co-founders, Ron Weiss and Daryl Irvine. And with that, I'll stop here and take any questions.
Excellent. Thank you, Jaspreet, and all of our three speakers here in track 2, what's next for RNA. So we're going to bring a couple of them back. One from the audience is cell-specific expression achieved with mRNA binding site engineering or by some other mechanism?
Yeah, thanks for that question.
Yeah.
That is true. So the cell type specificity, one of the mechanisms is by engineering of the right microRNA binding sites in our payloads.
Great. You also, Jaspreet, you mentioned towards the end of your talk that many messenger RNA oncology trials have not yet delivered promising results. And I wondered if you could expand on that. What is the consensus as to why the results have not overall been as promising as you would have liked?
I think it is a combination of a couple of factors. I believe that as is a standard in gene therapy, delivery still remains to be a big issue. Combine that with the fact that maybe some of the trials have been focused on targets which are yet to show any promise in the clinic by any other means too. So as an example, cancer vaccine, I think the concept is not only novel, it's also theoretically, it sounds very promising, but I believe that even using other modalities, it hasn't been proven to show a lot of efficacy just because of the heterogeneity in the tumor microenvironment. That I would say is there are a couple of reasons why maybe mRNA drugs in oncology haven't shown a whole lot of promise.
Daniel, can I throw that question to you as well? I don't know if you're doing a whole lot of work in the oncology space, but you may have thoughts on that.
Well, so I agree with the statement that cancer vaccines in general have been challenging, and so it's not unique to messenger RNA. It's a challenging application. I personally am more excited about the application of RNA medicine in other spaces, including genetic disease, genome editing, and of course vaccines, where probably we've already gotten injections. But I think it's just a matter of time, and I also agree that the delivery systems continue to improve, and I think we're going to see more effective systems targeting delivery to the right tissues, and we will get some of these to work.
Yeah, great. Lior, welcome. Thank you for joining us. Hope you can hear us and we can hear you. We got an early question during your presentation from Nada in our audience who wants to know, do you get an innate immune response?
The activation of the innate immune system usually happens when you use a mRNA that contain uridine. The big discovery of Carrico and Weizmann was in 2008, describing in molecular therapy that the use of pseudouridine, one of the nucleotides, one of the uridines we have in our body, in higher percentage, over 50%, leads to ability to avoid recognition of toll-like receptor 7 and 8 of the innate immune system, the activation of the innate immune system. A good demonstration happened now during COVID as companies like CureVac and others use uridine-based mRNA and use the same sequence, same delivery method as BioNTech and Pfizer. Same sequence, everything is similar. Only one thing is different. BioNTech and Pfizer use modified mRNA, which is the pseudouridine, one methyl pseudouridine to be more specific, while CureVac have used only uridine. And one of the fundamental problems with the study that CureVac have seen is that when you go above a certain amount, if I recall correctly, it was above 5 microgram, you can get some neurological side effect. However, as you know, when Moderna and BioNTech went over to 30 and to 100 microgram per injection, indicate again the fundamental importance of the use of pseudouridine. And our work, starting in 2010, when I was a postdoc in Derek Ross's laboratory with Ken Chen and Bill Poole, we were one of the first of using this modified mRNA, which the whole idea is to prevent the activation. So 1 gene or 7 gene, it doesn't matter. Both of them have the same structure, so none of them activate the innate immune system drastically. And recently we have heard just yesterday a good result from work of Moderna using the same modified mRNA in the cardiac space using the VEGF modRNA, which shows again the importance of using modified mRNA over mRNA to prevent the activation of the innate immune system.
Let's just pose for the panel and get their thoughts on the main theme of this track. What's next for messenger RNA? You've all sort of addressed it during your various presentations, but Jaspreet, let me start with you. I wonder what you think is going to be what looks like the most exciting avenue in mRNA research, or your biggest hope for the field, maybe over the next 5 years or so.
Yeah, I think, I share Dan's thoughts that, with delivery, innovation and delivery can really help us, not only avoid, off-target tox, but also provide, better therapeutic index for, our drugs. What has been encouraging so far, at least, one of the points that I made in looking at the different mRNA drugs in different stages in the pipeline is that they have been safe. That is great. Now we just have to find better targets, have the right approach, and hopefully, that can provide, better therapeutic index in, multiple different diseases, not only oncology, but genetic diseases as well.
Great. Dan, I want to put the same question to you.
Well, 5 years seems to go by pretty quickly to me. In the short term, we're going to see new drugs for vaccines for different diseases. But, when I look further out, I am very excited by the application in genome editing space in the treatment of monogenic inherited disease. I think cancer is an exciting area. I think it's really the tip of the iceberg, and what is going to lead the way is better and better delivery to new tissues and new organ systems.
Yeah. How do you, there's been a lot of progress, as we heard from Greg Newby, I'm sorry he couldn't join our panel today, in base and prime editing, which he didn't have time to really talk about. How do you gauge the progress that's being made using those technologies?
Yeah, it's almost like you can't read another journal article without a new CRISPR system coming out. So frankly, it's been remarkable. As exciting as base editing was, then we see prime editing, and then we see these new forms. And, it's still really at the preclinical stage. And so I think we're all holding our breath to see how it ends up working in people. But, again, based on the progress of messenger RNA on the whole, and the genome editing field in all these directions, I think it really looks promising.
Yeah. Lior, let me ask you, can you unmute and give us your big picture prognostications?
I'm looking forward in the coming years for more self-specificity type of treatments, which will be focused on different diseases. I think that the proof of principle that COVID has gave us and focus on vaccination only will emerge into more a belief in the power of mRNA therapeutics in different organs. And I would be very curious to see how it works. However, delivery is still number one issue, and self-specificity is the other one that needs to be faced. And I think that, again, in preclinical trials, people are now developing new technologies, like was described today by Strand Therapeutics. And I think that others are doing important work in the preclinical space to trying to find new tools that we can use. And hopefully when that will go into large animals and hopefully into human, that will be very interesting to see how it all comes together. It's going to be a very interesting decade for mRNA therapeutics, that's for sure.
Yes, yes. I'm going to squeeze in two quick questions from the audience. Ashish asks, what are some strategies that can be used to deliver mRNA products, preferentially into the brain via IV? Would anyone like to answer that?
Well, I would say a needle is a good way. The brain is a really challenging area. I think there's been some pretty exciting technology about crossing the blood-brain barrier, but in the context of messenger RNA, there's still some room to go.
Yeah, yeah.
I can agree with Dan, at least like in our hands, and we are working on this space over a decade, and independently in my life for over 7 years. The only real data that I saw in brain delivery was by direct injection. We had an R21 that we got for intranasal delivery to the brain of peptide. It was a collaboration with Ala Aroni Nicholas here in Mount Sinai, and we used intranasal delivery to trying to see if we can upregulate peptides in the brain. We partially had some positive results, but again, nothing like too exciting. I think the key is really direct injection right now.
Dan, let me start with you on this one, and anyone else can chime in. Sanjay wants to know, is there any work going on where mRNA release from the nanoparticle can be prolonged so that its therapeutic effects can be further extended?
Well, there certainly is thinking around how to get extended RNA effects, and it doesn't necessarily have to be released from the nanoparticle. The nanoparticles can be in something that are released over time. There are issues with formulation stability and payload stability that need to be addressed. But I think it is an interesting direction. And one point of reference, although it is technically different, is in small RNAs we've seen things go from lasting weeks to lasting months and perhaps even a year. And so who knows? We may see the same thing with messenger RNA.
Okay. Any other final thoughts from the panel? All right, I'm going to leave it there in the interests of time. I do want to thank Jaspreet, Dan for coming back to us, Lior. So thank you all very much indeed. That concludes track 2, what's next for mRNA. I want to thank all of our speakers, and we'll let them go now. The closing plenary session will begin on the main stage in just a couple of minutes at 3:35 p.m. Eastern. So we invite you to move over to that session now, and we'll see you on the main stage for the final 25 minutes of mRNA Day. See you there.